All right, thanks very much to Troy and Megan and all the other organizers for putting this on. It's great to be in a place where people know what you mean when you say Anchialine. Very weird feeling. And I'll try to keep this short because I know we're running late. Yeah, so in our lab, we study a lot of very diverse evolutionary processes, everything from aging to speciation. We do a lot of mutation and molecular evolution work as well. The thing that unites all of these really disparate projects in the lab is that we tend to think about how mitochondria influences all these things, both the physiology and the function of mitochondria, and also their genomes, their genomics, and their evolution. But we have this weird stepchild research program in the lab, which is our Anchialine research program. And so we don't have anybody in the lab whose dissertation is focused on Anchialine biology. It's the system that I did for my PhD, and I love it, and I've tried to keep it going and force people to do side projects. So I'm going to talk today about some of those side projects that we've recently been working on, and then how we're trying to bridge this gap and kind of get mitochondrial stuff going in the Anchialine ecosystem. So everybody has been talking about these habitats. If you go to Hawaii and look at one of these Anchialine habitats, you'll see something different in every habitat, which is something that somebody previously said, but this is a really cool, pristine one, Hawaii Bay on the south of the Big Island. And of course, you see in the foreground all these really charismatic little red shrimp running around that we've grown to love here and over the course of the conference. So, as Scott mentioned earlier today, it's not really appropriate to think this as a single thing across all the islands, but there are all these different genetic lineages of Halocaridina found across the islands. But this work, was based solely on mitochondrial markers, which is not atypical for population genetic work. Unfortunately, there's lots of reasons why what's going on with mitochondrial DNA is not necessarily reflective of what's going on in the nuclear genome. So there's this nice review by David Tells and Mike Brelsford, basically exploring different reasons for what we call mitonuclear discordance. So we would expect that mitochondrial haplotypes would fall out according to the nuclear DNA tree or your species relationships, but there's lots of reasons why we can see different relationships, either strong structure in one genome compared to the other, or maybe a strong signal in both genomes, but very different signals depending on the genome. So we wanted to explore this in Halocaridina. So a PhD student in the lab, Jess Sterling, did this using 2B-rad-seq. So I won't get into the methods, but this is a genome-wide sort of a restriction-associated DNA technique. And so we had samples from pretty much across the islands. This included some older samples that Scott gave us. And we were able to characterize, based on about 120 individuals, 25,000 unique SNPs that we could use for population genetic work. And this is essentially the results of the study. These are kind of pie charts showing the fraction of ancestry for each one of these populations that we looked at. And then over here we've got this structure or admixture type of diagram. Each vertical bar here is a single shrimp, a single sample, and the colors correspond to the fraction of nuclear ancestry for each one of those shrimp. So I didn't tell the program, how many populations of shrimp we thought were there. We said, based on the data, you tell us how many populations are there, and this is what it came up with. So the big sort of take-home message I want you to leave with is that what we see in the mitochondria is also kind of recapitulated in the nuclear genome. There is this very strong population structure based on the nuclear genome. It largely follows the same sort of pattern that we see in these single mitochondrial markers. But if you look more closely, there are some pretty interesting differences. So first of all, there were only 7 nuclear lineages based on the nuclear data versus 8 mitochondrial lineages that we've identified previously. There's a cool site up here, KBP, which we'll be headed to in a week or two. It's interesting because it has actually two different mitochondrial haplotypes, one this brown haplotype versus this orange haplotype. And in KBP, we also see a mix of ancestry here in the nuclear genome, but it's pretty cool because it's the opposite proportions of what we see in the mitochondrial genome. So this population might be a really good place to look for signatures of mitonuclear coevolution, which is something that we're really excited about in the lab. And then also, you'll note, it's kind of weird from my perspective, that some of these populations for sure have a lot of mixed ancestry, and so there might be nuclear introgression or nuclear hybridization happening across maybe a wider range than what we see in the mitochondrial genome, which is pretty cool. So you can see this most notably kind of going from the west coast up towards the east coast of Hawaii, where we get sort of a more gradual blending of these two different lineages. All right, you can also make a phylogeny out of these kind of data. We have a pretty nice clade for each one of the islands, the only exception being these individuals from Hilo that are grouping more with Oahu, and so we're trying to explore that a little more closely. As Scott mentioned, some of the phenotypic differences that we see across these populations are in coloration. Some lineages tend to be more darkly colored than others, but yeah, it's pretty interesting. So just to conclude this first part, there are these genetically distinct lineages of H. rubra according to both genomes. So I think, being together and having, the world's experts on Halocaridina in this room, this is a good opportunity for us to sort of think about what are these things and what do we need to do in terms of conservation, right? We could call them lineages, we could call them populations, we could call them subspecies. They're solid species according to some species concepts, but some of these habitats or some of these lineages are only known from like a single anchialine habitat. And if that habitat gets filled in, we've lost that genetic diversity, or at least the ability to go sample it, fairly easily. We'd have to go drop a well and hope that the shrimp came back up. So it's something I'd love to have a conversation about. We're still finishing up this project. We're sticking around on the islands to get some more samples. If you have cool shrimp samples from unique sites, we'd love to include them. Hopefully this will be complete in the next year or so. One of the limitations of doing this kind of approach is that all these SNPs across the nuclear genome are from anonymous loci. So we can't say whether particular SNPs that vary from place to place are linked to maybe some sort of functional trait based on a gene that they're nearby. And that's mainly because we lack a really good reference genome for H. rubra. So that's something else that we're trying to put together in the lab. Chase Smith, a postdoc in the lab, has been doing these long-read PacBio hi-fi sequencing experiments to try to piece together the Halocaridina genome. We were able to largely find all these reference Busco genes, but there was this really high rate of duplication for them. And basically that was the tip of a very problematic iceberg in trying to assemble this genome. It seems like there's issues with ploidy, there's high heterozygosity, there's really big regions of repeats and duplications and stuff. So we're a long ways away from assembling a great Halocaridina nuclear genome, but we're throwing sort of all the new technological tricks at it. So we've been doing dovetail OmniC data. This uses chromatin information to link particular reads to try to get a better assembly. And this graph is just showing that, yeah, that helped out a lot, but we still have a lot of mess going on here. So we're still throwing other tricks at it and more money. If you're excited about a Halocaridina genome, talk to me and I can tell you how you can help us out. We're also interested in the physiology of these shrimp. So we did this cool recent experiment. This was led by a high schooler in the lab, Hilary Perales, but she's not here. She worked with Kendra, who is here, so you can go talk to Kendra about this, where we looked at metabolic rate of all these different lineages. And what we found is that, yeah, there might be some differences between particular populations, but in general, they all show, fairly similar resting metabolic rates. And this is kind of interesting because there's so much variation among these mitochondrial genomes. This is a little movie that Scott put together a while back, showing that there's partial gene duplications in some in particular lineages, which is pretty interesting for animal mitochondrial genomes. But it doesn't seem to be related to any sort of baseline functional differences in metabolism. Again, we like mitochondria, so something else that we've been doing is looking at mitochondrial function. One of the ways we do this is to isolate intact functional mitochondria and then do this sort of respiration kind of experiment where we add different substrates and inhibitors to turn on different parts of the electron transport system. If there are any fans of rotenone in the audience, it's one of the things we add. It's a complex 1 inhibitor, so it can tell us like how much complex 1 is contributing to respiration. But yeah, we're interested in using this in Halocaridina to see if there's differences among the genetic lineages in different environments, different stressors, things like that. Right now, these data were based on a pooled sample of like 15 shrimp, so we're trying to get this down so that we can do it in individual shrimp. And then also, we're interested in thermal adaptation in these shrimp. So this is a project that's been led by a couple undergrads in the lab, Alice and Mary Angel. Eric has been helping them. He's got a really cool poster out in the lobby showing this, but it seems like there are some cool thermal differences between the different lineages, maybe having to do with like volcanic activity and geothermal activity and stuff. So again, check that out if you're interested. And then finally, as Scott and Nelson have been talking about, we're also interested in the microbial ecology of these habitats and also the gut communities of the shrimp themselves. So again, Scott and Nelson talked about this paper. Jess is going to talk right after me about another really cool project looking at microbial ecology, so I don't want to steal their thunder. But you can see, kind of like Scott pointed out, we do have these really distinct microbial communities based on different kinds of samples. So again, something that's really cool and needs to be explored further. So again, I just want to thank the organizers, our funding sources. Again, everybody in the lab gets roped into working with the shrimp at some point in time. And then while I'm answering questions, I wanted to share some AI-generated art that has to do with Anchialine Ecosystems. So I'll open it up to questions.
Hello, Justin. Any time for one or two questions?
Yeah, Liz.
What do you attribute to the high duplication? Because also in the divided transcriptomes, the majority of the assembly is high duplication versus the single copy.
Yeah, so it turns out crustaceans have this really weird somatic ploidy issue. That basically, like as cell lineages divide, they can undergo independent polyploidization. So if you sequence like an entire shrimp, you can end up getting, some cells that are like octoploid, some cells that are diploid, some cells that are tetraploid. And that's on top of just the inherent difficulties of working with shrimp genomes. It shouldn't be too big of an issue with the transcriptomes though. So I don't know, we should talk more about that later. Because we have transcriptomes for Another quick question, maybe? Yeah.
So you geneticists, when you compare like the results from mitochondrial DNA to nuclear, does there tend to be a pattern, like you usually result in more lineages with mitochondrial DNA?
Yeah, so there's lots of reasons why you can see different patterns. It would be simplifying it to say there's a single general pattern that we see across all animals. But in general, you do often see like stronger population structure with mitochondrial DNA because in animals there's a higher mitochondrial mutation rate versus a nuclear mutation rate. So that's like one general pattern. But there's been lots of case studies where things are the opposite or different. So the important point is you need to look at both genomes in order to get the full story of what's going on with the animals themselves. Thanks.
Great, thank you.